Right. So yeah, I'm going to talk about extracellular vesicles from milk because that is the type of studies that we do here in Utrecht. And I'm going to talk about these EVs, and I'm calling them mother's little secrets because they are so tiny. And first, before I start, I need to acknowledge where our funding is coming from. So we get a lot of funding from the European Union and also from the Dutch government, but we also get funding from the industry, for instance, nutrition research, or a Dutch dairy company called Friesland Campina. And obviously, we are very grateful for the milk donors for providing us samples to work with. Before I go into it, I thought it would be a good idea to explain to you maybe what EVs are. I think some of you already know what EVs are. But when we consider a cell, it can produce small extracellular vesicles via different pathways. I'm going to get out the pointer. So, and there are several subsets of EVs that can be formed by cells. So in multivesicular endosomes, you can get the formation of particles that we call exosomes. They can be released by cells. And there are also the microvesicles. They are from the plasma membrane. And once they are released in the extracellular milieu, yes, we call them extracellular vesicles, and that is because we don't exactly know where they are from. Oh, now I'm hearing an echo. Do you hear an echo as well?
Yeah, a little bit. Maybe, oh, because you're unmuted.
Yeah, all right. So we call them extracellular vesicles because, yeah, we cannot distinguish the different subsets. Oh, today it's really a mess with my computer, and yeah, all right. So what are these extracellular vesicles? So they have a bilipid membrane, and they contain, for instance, various cargoes like proteins that can be in the membrane, but also on the inside. They can have metabolites, amino acids, and they're well known for carrying a lot of genetic material, especially microRNAs. And these extracellular vesicles are used by the producing cells to communicate with the recipient cell. And that recipient cell can then interact via also various ways. So for instance, you can have interaction on the surface via receptors and ligands, and then signaling can occur. But there can also be uptake of these extracellular vesicles via various routes. Or there can even be fusion of the EVs with the recipient cell, and then the intercellular content gets released into the recipient cells. So they're really a mode of intercellular communication, and these EVs are found in all bodily fluids, and also all cells produce EVs. So what happens then during breastfeeding? So we believe that the mother communicates to her infant via her breast milk, and as a cell biologist, I look at milk as a very complex biological fluid with various structures in it. And I look at things from a perspective of size, for instance. So in my opinion, when you look at cells in milk, they are the biggest structures you can find next to the milk fat globules. Then we know there's obviously a lot of calcium micelles in there that also have these colloidal structures. Loads of proteins, obviously, that we find in the wave fraction, maybe some microbiome there. We don't know maybe how much that is. And then finally, we have our extracellular vesicles. And what you can see from this cartoon is that these EVs overlap in size with other milk components. So that's potentially an issue when you want to separate them, but I will talk about that later. And what we sort of envision is normally when you think about food and the uptake of nutrients, we talk about the gut a lot. But you could also consider that the first entry point of the milk is in the oral cavity. And that is also a very important immunological and mucosal site. And as we use our EVs in our lab, just freshly isolated out of breast milk, we do not expose them to a lower pH or enzymes. So we need to take this into consideration when we translate our in vitro work to what would be going on in vivo. So that's why we say maybe something that we see in our lab goes on in the oral cavity. And when we look in the oral cavity, you can find a lot of immune cells beneath the epithelial barrier. So these can be innate immune cells, but also cells from the adaptive immune system, also T cells, which I will go into today. So how then do you separate these milk EVs from other milk components? That is quite a lengthy protocol that we use in our lab. So we start out with fresh breast milk, and this milk is obviously very complex, and we do a first centrifugation step that allows us to pellet any cells, but also getting a cream layer that contains the milk fat globules. So we can separate those, and then we get a milk supernatant we can store for long term. And then what we do is we do 2 additional centrifugation steps at higher speeds to get out cell debris or apoptotic bodies. And then we end up with what we call 10,000 G supernatant. This supernatant we put on top of a gradient with something that is called OptiPrep, which has a high density in the bottom and a lower density in the middle of the tube. We load our milk supernatant on top of that, and then we start spinning overnight at 192,000 G. So this is super fast. And what this does is that anything that is soluble, like antibodies, they will not go into the gradient. They don't have enough mass to be pushed into that gradient, so they will stay here. The EVs, they have quite a lot of mass. They will get pushed into the gradient thanks to the G forces. But they have a bilipid membrane and a cytosol. So at a certain point, they get stuck in the gradient. And we call that floating. So they float at their density. And larger complexes that also have mass, but don't have a bilipid membrane, go to the bottom of the tube. So in the EV field, this is considered one of the golden standards of EV isolation. So we take out the fractions that contain the EVs. But at this point, they are an OptiPrep, which we cannot use for our in vitro cultures. So we exchange this OptiPrep for any type of buffer that we want by using size exclusion chromatography. And in the end, we get our EV sample. Now, the thing is that if you want to study a biological effect of any sample, you obviously will need a control. And we decided we wanted something that comes from this milk background, but we thought we cannot use this 10,000 G milk directly on cells because it's not culture medium. So we just decided to repeat this entire step to get a procedural control, which maybe still contains something of the milk matrix. But our hypothesis is that we have purified milk EVs and we have a sort of empty procedural control that we can use alongside our assays. One other important thing to mention is that we start out with 6.5 ml of milk supernatant, and in the end, we get 7 ml, for instance, that's why it's pink, in culture medium. So we dilute out our sample a little bit, but we don't try to lose a lot of EVs. So we try to use the EVs in the same concentration as they are in milk. So one of the things that you can do is you can characterize your milk EVs. In this case, we looked at the particle concentration. As we expected, we find a lot of EVs in our EV samples and hardly anything in our control sample. When we look at the sizes of the EVs, we see that this is from 5 individual donors. They are around 200 nanometers in size, which fits the literature. And we can do Western blotting on markers that are specific for EVs, which are these first 4. So they are enriched in EVs, and we don't find them also in our EV depleted sample. Interestingly, we also took along lactoferrin, which is a very abundant milk proteins, which we find in all samples. So they are not specifically unique for milk EVs. And it also shows that this EV depleted sample is not completely empty. It still contains some milk stuff, but probably not many EVs. So it's actually, in our point of view, a very good control to take along. So one of the things that we did was a proteomic analysis on our isolated EVs. And the first thing that we did was comparing this proteome of the milk-derived EVs to the proteome of milk that had already been published. And I was actually surprised to see that we found over 600 proteins that no one had previously identified. And I think these were over 30 studies that were done in the past. And that probably has to do with our isolation procedure. So we get pure EVs, and doing with the proteomics that we get rid of very abundant proteins. So we go into depth much more. And then we started comparing the unique proteins of the milk proteome to the unique proteins that are only found in the Milky V's. And by doing Go analysis, what we saw is actually Go terms were enriched for Milky V proteins that had to do with signaling, cell adhesion, cellular communication, cell growth, and maintenance, while the proteins or the Go terms enriched for the proteins uniquely identified in milk, its complement, cytokines, chemokines, and also metabolism. So this would suggest that these proteomes have really a distinct function and that the proteome of Milky V's do something different than other milk components. Well, also one of the questions that we had is, when you look in milk, who is producing what and who could be the potential producers of the Milky V's? So what we did is we linked proteins to specific cells, also doing a Go analysis. And interestingly enough, we found a lot of immune cells linking to the Milky V protein and not so much for instance, memory epithelial cells or adipocytes or fibroblasts. So these data suggest that immune cells might be the producers of Milky V's and not so much the epithelial cells. And in another analysis, looking at the interaction possibilities of Milky V's with receptors to recipient cells, we came to this model where we see a lot of potential interactions between different cells. And today I'm going to focus on the T cell. So we can see that T cells might be the producers of Milky V's, but they can also maybe be targeted by Milky V's produced by other cells. So then the question becomes, how can Milky V's then influence T cells? So as I showed you, Milky V's can interact with a variety of immune cells that could be potentially present in the oral mucosa. And what has already been shown is that Milky V's can cross physiological barriers. So if the T cells are underneath the epithelial barrier, maybe in theory, these EVs can reach these T cells. So an in vitro assay that we run in our lab is that we culture purified CD4 T cells that we obtain from adult donors from blood. And then we stimulate them with antibodies directed against CD3 and CD28, which are receptors on the surface of the T cells that are used normally during immune responses. So we trick the cells into becoming activated and becoming part of sort of an immune response. And then we culture them with or without EVs or with the EV depleted control. And then we have a readout that we can look for proliferation and cytokine production, because that is what these T cells do when they become activated. They start dividing, and for instance, they produce cytokines. So here you see a flow cytometry plot of the cells that we cultured. They are stained with a dye, and if they are in medium without any stimulation, they just sit there and they don't do anything. If you stimulate them with the antibodies, part of them become activated and they start to divide. And what happens is when every cell division occurs, the dye is transferred to the two daughter cells and the dye is diluted between the two daughter cells. So you get a dilution of the dye. So this is an indication of proliferation. And what we actually see is that if we culture them in the presence of these EVs, there is no proliferation, while with the EV depleted control there is. And also if we quantify this in a bar graph, we see the same thing. So that's actually very interesting. And we were not really sure what to expect, but it seems then in this case that activation is suppressed in the presence of these human milk-derived EVs. And the same is true for the cytokines. So in the presence of EV depleted control, a lot of cytokines are being produced, but not in the presence of EVs. And in additional experiments that we did, we also saw that these cells remain naive and not become memory T cells. So there is sort of a brake system that is in play when these EVs target the T cells. So how does this work? Well, one of the things that we used, again, in this case was the proteomics, to look at the proteins that we identify in the Milky V's. And for instance, by looking at protein interaction networks, we saw that there were a lot of proteins that could potentially form complexes, which are also involved in various pathways, for instance, proliferation and signal transduction. And this could potentially explain what is going on in the T cells. And I will go into that in the next example, because we need to take one step back now, and we're going to repeat the T cell experiments again. But in this case, we're going to look at the sensitization status of the mothers, because we know that allergies can influence milk composition, and it might also affect allergy development in infants. So we repeated our experiment again with these purified CD4 T cells, and then we looked for proliferation and cytokine production again, but also the upregulation of activation marker and also transcriptional regulation. And in this case, we isolated EVs from mothers that we defined as allergic or non-allergic. And in this particular study, we said, well, mother in our case, or in our belief, would be non-allergic if we could not identify any specific IgEs, or if they had a very low total IgE count. On the other hand, allergic mothers would have a higher total IgE count, or they would have specific IgEs, for instance, against house dust mite, pollen, or dander. So when we compare them, so first of all, I'm showing you the non-allergic donors with the proliferation. With the EV depleted control, we see a lot of proliferation of the T cells as we expected, because we also saw that in the previous experiment. In combination with the EVs, this proliferation is halted. And what we saw is that when we use milk EVs from allergic mothers, they're still the same pattern, but there seems to be a small shift. And this shift was even more apparent when we looked at activation markers. So there is a significant difference between the EVs of mothers that are allergic and non-allergic in the upregulation of activation markers. And the same thing we saw for cytokine production. Even more so, for instance, we saw for IL-6 that IL-6 was the same or even higher when EVs from allergic mothers were added to the T cells compared to the EV depleted control. And the same was seen for some transcription factors or factor involved in cell signaling or in cell division. So in the presence of milk EVs, the stimulated T cells become activated, and the inhibition of T cell activation seems to be less with milk EVs from allergic mothers. So then again, the question is, what is the mechanism of action? So we went into the cargo again of these milk EVs to see if we can find anything there, because that is what these milk EVs are transferring between the producer cells and the recipient cells. So we look at the proteome of these milk EVs, and we also look at small RNAs, in this case, the microRNAs. And what we saw is that there was a small difference, or there were a few proteins that were different in abundance between non-allergic and allergic donors, and even more so for the microRNAs. So it seems that on in this case, these two cargoes, that especially the microRNAs seems to be affected by the allergic status of the mother. We looked at the targets of these microRNAs and the proteins, and then we did a Go analysis on these targets to see if it didn't make any sense. And what we saw from the Go analysis is that many Go terms were significantly different. And these Go terms were, for instance, involved in T cell activation or cell cycle, cytokine production, or signaling pathways. And what we also saw is that, that is depicted here in pink, that especially with milk EVs from non-allergic mothers, there seems to be a lot of regulation, and not so much with the milk EVs from allergic mothers. Although in this case here with the PI3 kinase pathway, it seems to be the other way around. So the next thing, because the thing with the Go analysis is you can get your Go terms, but you don't know yet who is doing what, like which proteins or which microRNAs are working in these T cells. So that is something that I'm currently working on. So this is work in progress. It's not completely finished. What you see here is basically a schematic representation of a T cell with the cell membrane, with the T cell receptor, with the CD28. And you see signaling cascades that lead to changes in these T cells. And what you can see is that In pink is the cargo from the non-allergic donors, and in blue is the cargo of the allergic donors, that this cargo can act anywhere in the signaling cascade. So it's not just on the top of the signaling, on the start of the signaling, or all the way to the end of the signaling. No, it's everywhere. And what you also see is that there's more pink than blue. So there seems to be more regulation by milky V's from non-allergic donors. And we have the idea that several downstream factors are being differentially affected by these milky V's. So that is also something that we tested on a messenger RNA level. So that is basically the validation of this model. And what we see when we look at the co-stimulatory molecules, it's basically the same pattern as I showed you previously. So when you look at the EV-depleted control, it gives a higher messenger RNA level compared to the EVs in the non-allergic donors. And with the allergic donors, there's not much regulation going on anymore. The regulation seems to be lost. And we also see that for all types of different messenger RNAs that are leading to growth factors, cytokines, and you name it. So this seems to be a very broad process. And yeah, interestingly enough, there is a difference then between mothers that have a different health status. So to conclude, we believe that the oral cavity should not be overlooked, and there might be already a lot of immune regulation going on there. What we see in our models in vitro is that human milky V's inhibit CD4 positive T cell activation. When we look at the cargo, we see differences between microRNAs and proteins, and we believe that this leads to differences in signaling cascades within the T cells. Other important things to mention, obviously, is that milky V's do not only interact with the T cells. There are a variety of other cells, including immune cells, but we also know epithelial cells. As I showed you, the allergic status seems to affect the cargo and the function, but in this case, we've only looked at the microRNAs and the proteins. It could be that other cargo is also responsible for the effects that we see, so like the lipids and the metabolites, but also other types of RNAs. As I said, I'm currently working on the pathway analysis. It's not completely done yet, but we can get a reasonably clear picture already at the moment. And what we will also do is we follow up on the children that, that we need to see whether or not allergic mothers, the breastfeeding from the allergic mothers did something to the health of the infants. So finally, I would like to acknowledge all the people that collaborated in our research, and especially from the team in Utrecht with Marka Waube and Marije Klein-Jan. We've been doing this research for many years, and I hope we can still proceed doing those researches for many years. So yeah, that was it. I'm happy to take on any questions.